Hello, good morning, students. I'm Dr. Bhatt. Good morning, students. I'm Dr. Bhatt. Teaching experience and research experience last 20 years. And I'm specialized in biotechnology. I have 5 patents and more than 150 papers. And I'm going to teach you the Unit 1 molecular interactions. As you can see, there are various kind of molecular interactions in various biomolecules. For example, DNA, tRNA, and there is interaction between DNA and protein. This is also kind of very good example of uh, molecular interactions. Bilayer membrane protein. This is also going to be part of your molecular interaction. And also there are various uh, proteins and molecular structure such as amylose, cellulose, collagen, amyloid, flagellar motor that is going to have molecular interaction. Ribosome you can see in the 3D structure. Sorry, molecular interaction, ribosome, very good example and often asked in the examination. So if you'll see what is the core of this molecular interaction. Core of this molecular interaction is various kind of bonds, various kind of bonds like hydrogen bonds, salt bridges, and there are so many non-covalent interactions which are basically operative in the molecular interactions. And these non-covalent interaction you can see are the core of central to life. And these non-covalent molecular interaction is a thermodynamic process that follow common interaction pathway, right? So in biopharmaceutics, in drug molecular interaction, there are so many non-covalent interactions. And if you'll see, the core of biopharmaceutical research is basically a kind of non-covalent interaction, basically the protein-protein interactions. You can see here in the chart, there are various bonds types, and it's a very important to understand their dissociation energy. Dissociation energy is important because when bond is there, that is non-covalent, that is not linked, right? For example, you can see in the hydrogen bond, this is a two molecule oxygen and hydrogen. They are partially attached due to attractive forces, due to dipolar interaction, due to partial charged appear over the oxygen and hydrogen. So energy is very, very important, and all this energy-based interaction, energy is required for covalent formation, covalent bond formation. That is the very strong bond, 30 to 260 kilocalorie per mole. Iron lattice, 250 to 4,000 kilocalorie per mole. Hydrogen bond, you can see 1 to 12 kilocalorie per mole. Dipole, 0.5 to 2. London dispersive force greater than 1 to 15 kilocalorie per mole. Hydrogen bond is the most important. So we are talking about the hydrogen bonds. This is about 5 kilocalorie per mole and around 21 kilojoule per mole in water. In water, you can see the hydrogen bond, power of hydrogen bond increases. That's why the range is 1 to 12. The solvent is very important. If you are talking about the molecular interaction, then two type of things are very important. One is bond and molecules which are interacting, and where molecule is interacting. Molecule is exactly interacting in the solvents, and there are so many type of molecular interaction you must know, must be aware, right? And first one is intramolecular forces, and second one is intermolecular forces. What is intramolecular? Intra means two atoms together within a molecule, within a molecule. And this essentially forming the chemical bond that define the molecular structure and property. For example, ionic bond between the ions, covalent bond because of sharing of electron pairs between the atoms, and they hold the atom tightly, atom together tightly. Okay, metallic bond characterized by sea of electrons. Metals, two metals are free to move around metal atoms, right? And next is the intermolecular forces. What are the intermolecular forces? That are the weaker forces that occurs between molecules, influencing physical properties such as boiling and melting points, solubility, and vaporization. And for example, dipole-dipole interaction between the polar molecule, for example, hydrogen chloride. One molecule is positive, one molecule is negative. That's why they are attracted. And that's why they are called as the dipole-dipole interaction. Second one is the hydrogen bond, a stronger kind of dipole interaction. And when hydrogen is especially involved and making bonds with highly electronegative atoms like oxygen, nitrogen, or fluorine. You might be aware of highly electronegative atoms. They're very strong. They can attract, right? And they can interact. They can create a stronger dipole interaction. That's why they are able to make the hydrogen bonds. And next one is the London dispersion forces present in all molecules. These are temporary attraction caused by momentary fluctuation in electron density, and that's why they are able to grasp a kind of molecule. And London dispersal forces, especially, you can see they are present in all the molecules in different environments. So three type of intramolecular forces within molecule we have studied: ionic, covalent, and metallic. Between metals, metallic bond. Between atoms, sharing of electrons, covalent bond. Ionic bond, when electrons are transferred between positively and negatively charged ions, right? So I think it's clear. Intermolecular forces, weaker forces between molecules, and they exactly influence the physical property such as boiling, melting point, solubility, and vapor pressure, right? So you can see here is the picture depiction of uh, intramolecular forces and intermolecular forces. Intramolecular between the atoms. For example, you can see here in the green structure, fluorine and hydrogen, this is very strong. While intermolecular forces are weak, but because of they occurring multiple times, they are very, very strong. So exactly you can define molecular interaction. These are the forces that cause molecular atom to be attracted or to be repelled from each other. These interactions, also known as intermolecular forces or non-covalent interactions. So you might be clear now. What are the molecular interaction? Molecular interaction are basically intermolecular forces or non-covalent interaction. So, for example, you can see there are various course of occurrence in the biological system. That's why they are very important in the biological system. For example, ice melting, water boiling, carbon dioxide subliming, protein unfolding, RNA unfolding, DNA strand separation, and membrane disassembly. All these are involving non-covalent interaction. And I have told you, molecular interactions are thermodynamic process, thermodynamic properties. So enthalpy of molecule is very, very important. Enthalpy of molecule between two non-bonded atoms must be ranging from 1 to 10 kilocalorie per mole, 4 to 42 kilojoule per mole, where the lower end is comparable to RT, and the upper end is substantially weaker than a covalent bond. So they are stronger, they are not stronger, right, with other covalent bonds. They are weaker, right? Enthalpy 1 to 10 kilocalorie, either you can say, or in the term of kilojoule, 4 to 42 kilojoule per mole. Okay, for example, you can see egg. Have you seen the egg? And if you are boiling the egg, what is happening? Boiling around 60 degrees centigrade, causing denaturation of eggs. That is called as the denaturation of protein. And this is exactly the conversion of native state to denatured state, right? How it is happening? You are boiling, you are breaking the bonds. You are boiling, you are changing the molecular interaction. And different molecules, different bonds are broken. Now they are free to move. And that's why you are using the term denature, right? Native protein means aggregated proteins. They are the natural proteins found by the natural interaction, molecular interaction between the various atoms, various proteins, and that's why they are able to form the large assemblies. And how you can identify between the natured egg and denatured egg? Because of scattering of light. Because of scattering of light, they give you a white appearance. Next example is, hello. Next example is when you add lemon juice to milk, what happens? The pH drops suddenly and the protein denature and aggregate, right? So similar phenomenon you can see with the melting of DNA while running the PCR, okay? So pH drops, Y protein denature and aggregate. I want answer in the next session. Okay, so very simple phenomenon. pH disturbs the molecular interaction because these are non-covalent interaction, easy to break because of shifting in the pH, shifting in the temperature. Slight shift in pH and temperature can denature these proteins. I hope this is very clear to you. Next example is molecular interaction, how they are stabilized if they are not interacted with the bonds. They are stabilized because of various folding. As you can see in the pictures, there are various steps in the foldings, right? Native proteins always denatured when there is a shift in the pH or temperature. This is called as the perturbation, right? Perturbation. This is going to sorry, change the balance from folded state to unfolded state. So how this perturbation occurs? This perturbation occurs because of small change in pH, temperature, or single mutation. They can unfold a protein from native protein to unfolded protein. There are so many denatured proteins in the surrounding water molecule or any solvent molecule, and there are huge number of interactions. That's why there is a intramolecular interaction. Large number of intramolecular interactions results in the formation of folded, stabilized structure in the biological molecule, right? So there are points called the tipping points, which decides and which is the point where denatured and native protein get stabilized. So what is the denatured state? When these molecular interactions are disturbed because of slight shift in pH or temperature, there is a perturbation or denaturation or separation of these stabilized structure called as the denaturation. For example, unfolding a protein, unfolding RNA molecule, separation of RNA molecule, separation of DNA molecule. Okay, and disassembly and melting of the ribosome. So then interior region become exposed to the surrounding, and mostly these are the water solvents. Molecular interaction within native state or assembly are replaced by molecular interactions with aqueous surrounding. That's why they are denatured. They are denatured. Okay. So covalent bond, these are not the focus point, but primarily they are very strong bond and they are present between atoms in DNA in proteins. There are various non-covalent interaction, weaker interaction. For example, hydrogen bond, hydrophobic interaction, van der Waals forces, dipole-dipole interaction, London dispersion forces, ionic interaction between the ions, cation pi-pi interaction between aromatic molecules. And cation pi-pi interaction between the aromatic molecule, that's why they are very relevant to biological system. Hydrogen bond, between the protein structure, between the DNA, between the water. Hydrophobic, when non-polar molecule, interaction between non-polar molecule. Listen carefully. Interaction between non-polar molecule in the aqueous environment. When we are trying to dissolve some non-polar molecule like benzene, like any biological molecule in the aqueous environment, for example, hydrophobic proteins, right? Non-polar molecule that's not having any charge, right? They are neutral. Amino acid, there are various non-polar amino acid. If you are dissolving them in aqueous environment, what is happening? Hydrophobic interaction arises. And this is very, very relevant to protein folding and membrane structure, right? So van der Waals forces, these are short range forces that arise from temporary fluctuation in electron distribution, dipole-dipole, attraction between polar molecule with the permanent dipoles. Already I have told you these interactions. So protein-protein interaction is also very, very important for the fundamental and cellular functions. And these are the specific interaction between proteins, protein-DNA interactions, interaction between protein and DNA, which control gene expression as the cellular process. What are the importance of molecular interaction? They are very important in determining the physical property of uh, molecules. For example, melting point, boiling point, viscosity, surface tension, and other property of liquid and solids. They are the driver of various biological process. For example, DNA replication, protein folding, enzyme catalysis, self-signaling are almost impossible if these molecular interactions are absent. So simply, you can see molecular interactions are important in every area. Drug designing and discovery, for example, understanding the molecular interaction is very crucial for designing drugs that can selectively bind to target protein and modulate their. There are also important material science, Understanding designing new material, nanotechnology, these interactions are crucial in the designing and construction of nanoscale devices and materials. All right. So next, in the next topic, we will understand how the B form of DNA is stabilized because of molecular interaction at molecular level. Thank you so much. Have a nice day. Thank you.